On June 29, 2026, researchers reported a discovery that made one of the brain's most important memory proteins appear even more disturbingly viral than anyone had imagined. The protein was called ARC, and it was helping neurons package another protein into tiny membrane-bound parcels. The cargo was tau, a molecule that normally supports the internal structure of nerve cells but can misfold and spread through the brain during Alzheimer's disease and related disorders. Without ARC, far less tau entered those parcels, and its transfer between neurons fell dramatically in experimental models. The finding revealed a dangerous double life. ARC is indispensable to normal learning and synaptic change, yet the same ancient molecular machinery may be exploited when pathological proteins move from one brain cell to another. Memory and degeneration appeared to be using part of the same delivery system. This was not a metaphor invented to make biology sound dramatic. Under powerful microscopes, purified ARC formed rounded shells with a geometry reminiscent of retroviral particles. Biochemically, it behaved like GAG, the structural protein that viruses such as HIV use to build the protective core surrounding their genetic material. Yet, ARC was not an invading virus moving through the modern brain. The gene was already part of the animal genome, inherited from parent to child, copied into nearly every nucleated cell, and carefully activated by neurons when experience demanded lasting changes in their connections. The title of this story therefore requires one correction. ARC is not a complete prehistoric virus sleeping inside every neuron. It is a domesticated descendant of a virus-like mobile genetic element, probably a Thai 3-eater gypsy retrotransposon, captured by an ancestor of land vertebrates roughly 350 million years ago. That distinction makes the story more remarkable, not less. Evolution did not merely tolerate the genomic remains of an ancient parasite. It stripped away much of the original machinery, preserved a useful structural gene, rewired its control system, and recruited it into the biology of learning. Somewhere in deep evolutionary time, a selfish sequence that once copied itself for its own survival became part of the machinery that allows an animal to remember where danger appeared, which route led home, who could be trusted, and what happened yesterday. To understand how that transformation occurred, we must first understand what a memory physically is. A memory is not stored as a tiny image, sentence, or video file inside one neuron. It emerges from coordinated changes distributed across networks of cells and the synapses connecting them. When an experience activates a neural circuit, electrical impulses travel along axons and trigger chemical signals at synapses. Some connections become stronger, others become weaker. New structures may grow, receptors move across membranes, and patterns of gene expression begin changing inside selected neurons. In the mid-1990s, scientists searching for genes activated by neural activity identified ARC as an unusually responsive molecule. Exploration of a new environment, strong synaptic stimulation, seizures, learning tasks, and sensory experience could trigger ARC transcription within particular groups of neurons. This selectivity immediately made ARC valuable as a biological map. When researchers examine where ARC RNA appears after an animal explores a space or learns an association, they can identify portions of the neural population recruited during that experience. Once produced, ARC interacts with numerous molecular systems. It influences the actin cytoskeleton that helps shape dendritic spines, the small protrusions where many excitatory synapses form. It also regulates receptors that determine how strongly a neuron responds to the neurotransmitter glutamate. ARC can promote anti-receptor endocytosis, pulling receptors away from the cell surface. At first, that sounds like the opposite of memory. If learning strengthens important connections, why would a memory protein participate in weakening synapses? ARC participates in this balancing act through several forms of plasticity. It has been implicated in long-term depression, homeostatic scaling, synaptic consolidation, structural remodeling, and the maintenance or refinement of experience-dependent changes. Its effect depends on timing, location, neuronal state, and interacting molecules. Homeostatic scaling is especially important. If a neuron receives persistently excessive excitation, it can reduce overall sensitivity to prevent instability. If activity becomes too low, it can increase responsiveness. ARC helps adjust receptor abundance, so circuits remain capable of encoding differences instead of saturating. This means ARC is not simply a molecular glue that freezes a memory in place. It behaves more like a network editor, helping decide which synaptic changes should remain, which should be reduced, and how neuronal activity can stay within a functional range. Early experiments demonstrated how essential this editing can be. When researchers blocked activity-induced ARC protein production in the hippocampus, animals could still perform aspects of short-term learning, but the consolidation of long-term memory was impaired. ARC-deficient mice later showed severe problems forming durable memories across spatial, fear-related, and recognition tasks. They could often perceive, move, and respond normally in the moment, yet the biological trace failed to stabilize with the strength seen in animals carrying functional ARC. The relationship with long-term potentiation, a persistent increase in synaptic strength often used as a laboratory model of memory, proved more complicated. Some studies found that blocking ARC disrupted maintenance of potentiation, while later work showed ARC was not always required for hippocampal LTP under particular experimental conditions. A memory also changes after it is first stored. Retrieval can make a memory temporarily unstable, requiring reconsolidation before it persists again. ARC expression has been linked to both initial encoding and later reactivation, suggesting it helps circuits update old information rather than merely preserve an untouched record. This flexibility is crucial. A perfectly permanent nervous system would become trapped by outdated predictions. Animals survive because remembered locations, threats, rewards, and social relationships can be revised when conditions change. Forgetting and updating are not failures of memory. They are part of its adaptive design. For years, neuroscientists studied these roles without realizing why the ARC protein possessed such an unusual structure. Its amino acid sequence showed similarities to retroviral gag proteins, but the evolutionary meaning of that resemblance remained uncertain. The answer began inside the enormous archive of mobile DNA carried by animal genomes. Transposable elements are genetic sequences capable, or once capable, of copying and inserting themselves into new genomic locations. Many are ancient, damaged, and permanently immobilized. Others remain active in certain species or tissues. Retrotransposons copy through an RNA intermediate. A sequence is transcribed into RNA, reverse transcribed back into DNA, and inserted elsewhere. The long terminal repeat retrotransposons share evolutionary and structural relationships with retroviruses, although they do not necessarily possess the machinery required to move freely between organisms. A typical retro element treats the host cell as infrastructure for reproduction. Its proteins package or process its own RNA, protect it, and help create additional genomic copies. From the host's perspective, uncontrolled insertion can damage genes, disrupt regulation, or create genomic instability. Hosts therefore evolve defenses to silence these elements. DNA methylation, repressive chromatin, small RNAs, and immune-like surveillance restrict their activity. Across evolutionary time, a molecular arms race unfolded between sequences trying to spread and organisms trying to contain them. Most captured elements decayed into genomic fossils. Mutations accumulated because the host gained no benefit from preserving their original function. But occasionally, one component performed a task useful enough that natural selection began protecting it. This process is called molecular domestication or exaptation. A selfish gene is not consciously transformed, and evolution has no plan. Random variation changes the sequence, while individuals benefiting from the new function leave more descendants. Gradually, an invader becomes an inherited tool. The original insertion did not immediately produce human memory. Early nervous systems already learned long before ARC was domesticated. Fish without the tetrapod ARC gene can learn, and many animals rely on different molecular strategies. ARC became one influential addition to an already ancient plasticity system. An even stranger clue came from insects. Fruit flies possess ARC-like genes that also derive from retrotransposon gag, but evolutionary analysis indicates that fly ARC and tetrapod ARC were domesticated independently from different mobile elements. Evolution had reached for a similar viral tool more than once. In flies, ARC1 protein forms capsid-like structures, binds its own messenger RNA, and moves across the neuromuscular junction inside extracellular vesicles, influencing plasticity and development. The decisive mammalian evidence arrived in January 2018. Researchers purified ARC protein and observed that it spontaneously assembled into virus-like capsids. The particles enclosed RNA, with ARC showing a preference for packaging its own messenger RNA under experimental conditions. Neurons released ARC in extracellular vesicles, small membrane-bound packages used by cells to transport proteins, lipids, and nucleic acids. These vesicles protected ARC and its RNA cargo as they moved through the surrounding environment. When recipient cells took up the vesicles, ARC messenger RNA entered them and could be translated into protein. A gene inherited from a retrotransposon was still performing an echo of its ancestral function, building a shell carrying RNA, crossing a cellular boundary, and releasing genetic instructions. The discovery forced neuroscientists to reconsider communication in the brain. Classical synaptic transmission relies on neurotransmitters released into a narrow gap and detected by receptors within milliseconds. ARC suggested a slower channel in which neurons might exchange complex molecular cargo capable of changing protein production. But one crucial boundary must remain clear. ARC capsids have not been shown to package a person's memory as a transferable object. They do not contain the image of a childhood home, the sound of a voice, or the meaning of a learned word. The cargo observed in the landmark experiments included an ARC messenger RNA, an instruction for manufacturing ARC protein. A memory remains distributed across patterns of synaptic strength, neuronal excitability, gene regulation, structural change, and coordinated activity throughout a circuit. Calling ARC a packet of information is accurate only at the molecular level. The packet carries RNA sequence information, not the complete experiential content encoded by a neural network. The difference separates genuine discovery from science fiction. Another uncertainty concerns what ARC capsid transfer normally accomplishes inside a living mammalian brain. The 2018 experiments established the biochemical capacity for intercellular RNA transfer, but researchers are still determining when, where, and how strongly this mechanism contributes to learning in intact animals. ARC's established importance for memory may arise from several functions operating together. Its intracellular control of receptors and cytoskeleton is well supported. Its extracellular capsids may coordinate nearby neurons, glia, or synapses, but the physiological message and destination rules remain active areas of investigation. The capsids themselves are not identical to infectious viruses. ARC lacks the complete set of genes needed to replicate autonomously, invade an organism, copy itself uncontrollably, and spread between hosts. It relies on normal cellular transcription and translation like other host genes. These populations are often described as engram cells, neurons whose physical or chemical changes contribute to a memory trace. ARC is not the engram itself, but its rapid activity-dependent expression can mark cells entering the process and help stabilize the changes they undergo. Experiments that label and manipulate active neuronal ensembles have shown that reactivating certain cells can trigger behavioral recall, while suppressing them can interfere with it. Memory, therefore, depends on both the selected cells and the synaptic architecture connecting them. ARC helps convert selection into persistence. A burst of neuronal firing can recruit transcription, send RNA into dendrites, alter receptor trafficking, reshape spines, and modify the probability that the same network will respond again in the future. This is another reason the statement, "without ARC there is no memory," is too absolute. ARC is critically important for many forms of long-term memory in mammals, but memory is not produced by one protein, and short-term learning, or alternative plasticity, can persist when ARC is disrupted. A more accurate statement is that ARC provides a major route through which activated neurons convert experience into durable, adjustable circuit change. Removing that route severely damages long-term consolidation, even though the nervous system retains other mechanisms. The distinction matters medically. A molecule essential for healthy plasticity cannot be blocked casually. Any treatment targeting ARC would need to preserve learning while correcting harmful activity, an extraordinarily difficult balance inside a system where timing and location determine function. ARC has been investigated in relation to Fragile X syndrome, schizophrenia, Alzheimer's disease, and other neurological conditions, because many disease-associated pathways converge on synaptic regulation. These connections do not mean an ARC abnormality alone causes each disorder. Vesicles isolated from tau-expressing mice contained forms of tau capable of seeding aggregation in recipient cells. When ARC was genetically removed, the vesicles carried much less human tau and lost much of their ability to initiate those aggregates. In living mouse experiments, intercellular tau transmission was strongly reduced without ARC. ARC and tau were also detected together in extracellular vesicles isolated from mouse and postmortem human brain tissue, linking the laboratory mechanism with material found in the human nervous system. At first, the results suggested a simple therapeutic strategy: remove ARC and stop tau from spreading, but biology immediately produced a complication. Without ARC, pathological tau accumulated inside neurons, and early neuronal loss increased modestly in the experimental model. The study also relied heavily on engineered mouse models expressing disease-associated human tau. It does not prove that ARC controls Alzheimer's progression identically in every human brain. Clinical significance will require additional research involving human disease stages, natural tau levels, and long-term outcomes. ARC's context is exceptionally precise. During learning, a brief pulse in selected neurons may help reshape connections and coordinate molecular responses. During disease, chronic activation or abnormal cargo may redirect the same system toward maladaptive signaling or pathological spread. This raises a question researchers have only begun to answer. What determines what ARC packages? The protein can bind its own RNA, but its interactions may depend on RNA abundance, sequence, structure, binding partners, cellular stress, and the composition of extracellular vesicles. Another question concerns destination. Do ARC-containing vesicles move randomly, remain near active synapses, follow specific connections, or target cells displaying particular surface molecules? A delivery system becomes far more biologically meaningful if the sender can influence who receives the cargo. Researchers must also determine whether ARC capsids travel primarily between neurons or communicate with astrocytes, microglia, and other brain cells. Memory is not created by neurons alone. Glial cells regulate metabolism, inflammation, neurotransmitter clearance, blood flow, and synaptic maintenance. The 2026 TIU findings make these questions urgent because extracellular vesicles can protect cargo from degradation and potentially carry it through connected tissue. Understanding the packaging rules may reveal ways to preserve physiological signaling while preventing pathological transfer. ARC's viral ancestry may also inspire technology. Engineered capsid-like proteins could potentially deliver RNA to selected cells without using complete viruses. Because ARC is derived from an animal protein, researchers are investigating whether its architecture can be adapted into controllable delivery platforms. The human genome is filled with evidence of such conflict. Large fractions derive from transposable elements and ancient viral relatives. Most are inactive fragments, but some provide regulatory switches, protein coding sequences, or structural innovations that influence development, immunity, reproduction, and brain function. The transformation also challenges the idea that parasites and hosts occupy permanently separate categories. Once genetic material enters the germline and becomes inherited, evolution can renegotiate the relationship. What began as exploitation can become mutual dependence. Over hundreds of millions of years, that contribution passed through creatures radically unlike us. Early tetrapods, reptiles, nocturnal mammal ancestors, primates, and humans all inherited versions of a molecular system whose remote ancestor once served a mobile genetic element. A structure designed to protect RNA became a device for neuronal communication. A protein that once helped a sequence spread became a protein helping experience alter a brain. An ancient invasion became an inherited capacity for adaptation. Inside an activated neuron, ARC messenger RNA may be transcribed because something important has happened. The cell sends those instructions outward, changes the receptors at selected synapses, reshapes its connections, and potentially releases molecular parcels toward neighboring cells. Hours later, the electrical activity has vanished. The original sensory signals are gone. Yet the circuit is no longer exactly what it was. Its future responses have been altered by proteins produced during that narrow window, including a molecule descended from a genomic invader. That altered circuit is not the complete memory, but it is part of the physical transformation that allows the past to influence the future. Without such changes, experience would pass through the nervous system without leaving a durable biological consequence. Your genome, therefore, contains more than a record of your ancestors. It contains captured technologies from organisms and mobile elements that once challenged them. Some were silenced, some were broken, and a rare few were recruited into the machinery that makes complex life possible. ARC is one of those recruits. It does not awake inside every neuron continuously, and it does not carry personal memories across synapses inside microscopic viral capsules. Reality is subtler and more extraordinary. The gene waits under cellular control until activity summons it. It produces a protein that regulates synapses, supports long-term memory, and retains the ancient ability to form capsid-like assemblies and transport RNA between cells. Without ARC, the mammalian brain does not become completely blank, but many durable memories fail to consolidate normally. With ARC, neurons gain a powerful method for converting activity into controlled structural and molecular change, although disease can exploit that same machinery. The ancient parasite did not simply survive inside us. Evolution changed the terms of its survival. Its descendants became useful only when the animal remembered, adapted, and lived long enough to pass the gene onward. And every time a memory persists, every face recognized, every warning retained, every skill strengthened, the modern brain may still be using the repurposed architecture of an invasion that began roughly 350 million years before the first human ever had a past worth remembering.